Hello, it's Mr. Cho here, and today we'll be going through chapter 2 of your biology textbook, right? Movement of substances. Okay, so this is going to be quite a long chapter, so let's buckle up and let's get ready. Alright, so in this chapter, you'll be actually working with potatoes because this one it's a living cell, right? So potatoes are made from cells, plant cells, and we'll be using them for experiments. Okay, so there's a scenario where you are a chef, alright, or rather there's two chefs and they are cooking potatoes, and each of the potatoes need to be cut into 4 pieces, and each potato also needs to be soaked in water before cooking. So Chef Nurul and Farida took a potato each and scraped off its skin. Chef Nurul cut the potato into 4 pieces before soaking it in water for 1 hour. Chef Farida soaked the potato in water for 1 hour before cutting it into 4 pieces. Which chef would need to exert more strength to cut the potatoes? Okay, why do you think so? Okay, so using your knowledge about osmosis, what do you think? Okay, shout out your answer. Okay, the answer is actually Chef Farida. Alright, so because when Chef Farida put it into the water for 1 hour, osmosis has occurred, so some water has entered into the cells, and therefore the cells are what you call as turgid. Turgid means hard, so it should be harder for her to cut in the water. Okay, this is I mean, it's not going to be that much harder lah. It's just like maybe a minuscule, tiny little bit of strength more only. Okay, so let's begin the chapter. So what are we supposed to learn today? Today we need to learn that we should define diffusion and describe its role in nutrient uptake and gaseous exchange in plants and humans, and describe the factors that affect the rate of diffusion. Okay, so what is diffusion? Now imagine that you are reading a book, and then your sister spills her perfume in the living room. Okay, now over a very short period of time, you will start to smell that perfume, right? Why is that happening? This is because perfume, like all matter, is made up from particles. Okay, in this case, the perfume is made up of molecules. Right, so we call it as the perfume molecules, and these perfume molecules, they actually evaporate really easily from the surface, as in they will become a gas, and the gas particles will then have some kinetic energy. They will also be blown around by the wind in the room, and if the room is hot, it's going to move around. Then it moves around in all directions, and because we learned before in this idea of kinetic particle theory, how the molecules will bump into each other and move about. This will cause the gas to spread throughout the house until it reaches your nose, and then you will smell it. So the process in which this spreading out is called diffusion. Okay, so there's these 2 areas in the room, right? Let's call this area as point A, and let's call this area as point B. So what we see is that when the perfume is spilled, there's going to be a high concentration of the perfume at point A. Now what is concentration? Concentration refers to the amount of a substance within a fixed volume. Okay, so that means that within this volume, there is a certain amount of the perfume molecules. So there's going to be a very high concentration of perfume molecules here because you just spilled the perfume there. Whereas there's a low concentration of perfume molecules initially at point B because that's the air. There shouldn't be any perfume molecules there until it starts to diffuse over there. So the molecules at point A are high, and the molecules at point B are low. So this is what you call as a concentration gradient. There's a difference in the concentration between points A and point B. So if we were to represent that in a graph, it will look like this. Point A is high in the perfume molecules. Point B is low in the perfume molecules. So there is a difference. This difference is called a concentration gradient. Okay, so the definition of concentration gradient is the difference in concentration between two regions. Please highlight this sentence. So the particles of fluids such as liquids and gases, they possess kinetic energy, right? So they're constantly moving about, and because their movement is also random, these particles will diffuse down the concentration gradient and it will eventually become spaced out after a short period of time. So it's shown in this figure. There's a high concentration here. It will move in all directions across the room. So, to the left of the room, to the right of the room, up and down the room. It's going to spread out throughout the entire room. So this is where we get the definition of diffusion. Diffusion is the net movement of particles such as atoms, molecules, or ions from a region where they are of higher concentration to a region where they are of lower concentration. That is down a concentration gradient. Please put a big star on this definition. Definitions are part of your exams, unfortunately, right? You do have to regurgitate this out. Okay, so let's look at an example. All right, let's using a video. Let's watch how. Okay, but before we do that, let's quickly just show one more time, right? What concentration gradient means. So here you can think of concentration gradient like the example of a slide. Okay, I used to love going down to play slides, but nowadays the slides are pretty boring. They're not fun anymore. So yeah, when I was young, I would love to go up to the highest slide. Why? Because I will slide down the fastest. Alright, everyone knows that. In the same way, when you have a very concentrated area, you're going to have a faster rate of diffusion. So you imagine that there's a high concentration of the perfume molecules at one region and absolutely no perfume molecules at another region. Therefore, the molecules will go down the concentration gradient really quickly. Okay, not slide anymore. However, after a certain period of time, the concentration gradient will start to become less steep. So there's going to be maybe a medium amount of perfume molecules at one region and a slightly lesser amount of perfume molecules at the other region. Okay, so this will then take a longer time, a slower rate of diffusion for the molecules to move from one area to the other area. Okay, what else can increase the concentration gradient? Okay, there are two things. Firstly, temperature. Okay, so if there is a high temperature on one end, so like for example, if you have some liquids, right, and the liquid is hot, then the kinetic energy of the heat, right, will actually cause the particles to move around more quickly, and therefore you increase the rate of diffusion in hot water versus in cold water. And another will, of course, if you have like kinetic energy, so increase the kinetic energy of the liquid. For example, you have wind or you're stirring something, that also naturally would then increase the rate of diffusion. Okay, now let's show you this quick experiment about Skittles diffusion. Okay, let's watch it together.
Bearded Science Guy here, and today I'm going to show you how to make the Skittles diffusion experiment. To start with, arrange a group of Skittles around the edge of a plate. I found it works the best to have at least 2 of each color in a row. Now take warm to hot tap water and pour it slowly into the very center of the plate until it's just covering the Skittles. Almost immediately, you'll start to see the colors running toward the center of the plate. This is because the molecular structure of the dyes used on Skittles makes it easy for them to dissolve and then diffuse through warm water. To continue having fun with this experiment, try changing some of the variables, such as the temperature of the water or the type of candy you're using. Thanks for watching, Bearded Science Guy. If you enjoyed this video, consider some molecular structure of the...
Okay, so what do we just see? The Skittles contain dye molecules, and dye molecules are soluble in water. So when you put water on the Skittles, the dye molecules start to dissolve. And when they do dissolve, there is a high concentration of the dye molecules at the Skittles, right? Across all the Skittles, there's a high concentration. And where is there a low concentration? Right in the middle. So in the middle, there's nothing, right? There's no Skittle dye molecules. So it's what we call as extremely low, or even zero concentration gradient. So the concentration gradient is high from here to here. And therefore, there will be a net movement of the Skittle molecules from a region of high concentration down concentration, sorry, let me say it again. There'll be a net movement of the Skittle molecules from a region of high concentration to a region of low concentration gradient down the concentration gradient. Okay, so this is how you write a diffusion statement. Okay, so we saw in the video that the particles, right, of, okay, not the copper sulfate, but we saw the particles of the Skittle molecules, right, move from a region of higher concentration to a region of lower concentration in the surrounding water. And eventually, the particles will diffuse evenly throughout the entire liquid to form a solution. And because you saw in the video also that there were different dye molecules, right, different colors, they are actually going to dissolve and diffuse independently of each other. So what after a certain period of time you will see is that the colors will start to mix. You know, they will eventually just become like just one weird color, right? Because all the particles will just start to mix together. Okay, so that was what I call as diffusion without a membrane. So diffusion doesn't really need a membrane. Okay, but sometimes we do use a membrane, right? Even when there's diffusion, right? Just to prove a certain point or to do a certain kind of experiment. So let's look at this experiment. Here we have a permeable membrane. Okay, so the meaning of a permeable membrane means that most things can pass through it. It's not semi-permeable, it's not partially permeable. It's just like more or less fully permeable, right? The things can just quite easily move through them. And this permeable membrane, it allows both the solvent water and the solutes, the dissolved substances, to pass through it. So let's look at what we have here. Over here, we have a left side with a higher concentration of potassium nitrate in yellow. And on the right side, we have a higher concentration of copper 2 sulfate. Okay, so these are two different chemicals, right? So one is found on the left, one is found on the right. After a certain period of time, you will see that they will start to mix, right? The potassium nitrate will move towards the right, and the copper sulfate will move towards the left. Okay, so at the end, you will see a mixture, right? You'll see this mixture where both sides have roughly equal amount of the two different particles. Okay, so what has happened? The particles of copper 2 sulfate has diffused across the membrane to the left side of the beaker, and the particles of the potassium nitrate has diffused across the membrane to the right side of the beaker. And eventually, there will be an equal concentration of particles on both sides of the membrane. Okay, so there's this word called net movement. Okay, I want you to take extra precaution about this. Oftentimes, we may say, okay, if the particles move from this side, right, they move to the left side, and then the right side move to the left side. You know, once they have reached over there, they stop moving. Okay, that's not how reality actually is like. Even though the particles have an equal concentration, they are still going to move. Right, so what do I mean by that? That means that even though it has already achieved equilibrium, the word equilibrium means equal, the particles are still actually moving left and right. You know, sometimes this one is moving to the left, sometimes this one is moving to the right, and etc. etc. But when it says no net movement, it means that the overall movement of all the particles cancel each other out. That means that if let's say there's one yellow that moves to the left, then one of the yellow here will move to the right. And if there's one blue, then move to the right, then one of the blue will move to the left. Okay, so that means that if one is moving left, one is moving right, they cancel, right? The total number is still the same. Okay, so that's why it calls no net movement. Okay, likewise over here, when I say net movement of potassium nitrate particles, it means that yes, majority of the yellow dots are going to move towards the right. But that doesn't mean that 0 yellow dots can move back to the left. Okay, they can actually still move back to the left. Okay, so it's like majority goes over to the right, then sometimes they'll still go back a bit. So it's like a constant movement. Right? It's this idea of a constant movement, but eventually all these constant movements, they kind of like balance each other out. Okay, and that's why it calls no net movement. Okay, so that is diffusion through a permeable membrane. Now let's talk about this concept of surface area to volume ratio. We know that cells are extremely small, right? Most cells are extremely small. Why is that the case? This is because the cells need to have materials move across the cell membrane quickly. And we know that cells need oxygen, nutrients, and waste materials to move across their cell membrane fast enough so that the cell can stay alive. Okay, so there's this constant exchange of oxygen and nutrients into the cell and waste materials out of the cell so that the cell can stay alive. Now, the rate of movement of a substance across a cell membrane, it will depend on how large the cell membrane is. That is the total surface area of the cell. For two cells of the same volume, okay, so volume means how much space it takes up. So if they're the same volume, then the one with a larger surface area will allow for a higher rate of diffusion. Okay, so it's getting a little bit complicated. We'll watch a video to kind of simplify this. There's this thing about ratios, right? So let's look at different ratios. The ratio between the surface area and volume of a cell is called surface area to volume ratio. Okay, please spell out this whole thing correctly. I tend to say as a shortcut SA to VR, but when you write it for exams, you have to write it fully. Okay, so what is SA to VR? This is an essential concept in biology. The greater the SAVR of a cell, the higher the rate at which substances can move in and out of it. Let's look at this experiment over here. So we have a small cube, we have a medium cube, and we have a large cube. Okay, which of them do you think will have the highest surface area to volume ratio? Is it the small cube, medium cube, or the big cube? Okay, think about the answer. Just shout it out. Okay, if you said small cube has the higher surface area to volume ratio, you are correct. Why is that the case? Let's do some math. So, if we take the volume of a cube, alright, let's calculate the surface area first, since that's what the question is saying. If you calculate the surface area of a cube, we see that it's one of the faces of a cube is 1 cm square. There's a total of 6 surfaces in a cube, so that's 6 cm squared. And the volume of this cube is only 1. But if you compare this to the volume of the medium cube, the volume of a medium cube will be 2 times 2 times 2. That will give us 8. Okay, and then what is the area of this one surface? It's 2 times 2, right? So that's 4. 4 times 6 is, well, my math is bad, 24. Okay, so 24 and 8, right? So 24 and 8. So what's the ratio then? 24 divided by 8, that will be 3:1. 3 cm cube, 3 cm square is to 1 cm cube. Okay, so it's not as good as this, right? This is 6:1. That was only 3:1. Okay, and of course, if you do the last one, and you try and do it also, then you will see that it shrinks even more, which is worse. So the total surface area for the smaller cubes is higher. Let's watch a little video to help us understand that. Okay, so here in this video, we start with two identical identically sized agar blocks. We will then chop up one of the agar blocks into smaller cubes, right? Which technically also means that it has the same volume because it starts, these two cubes are the same volume. So you cut them up, and by cutting it up, you increase the surface area, the total surface area of the cubes.
Volume and the surface area. So what you should do is, when you do the lab, you want two of these. One you're going to keep the same, and the other one you're going to chop up into 8 pieces. The way that we chop up 8 pieces is you can make a cross on top and then slice it thick, wide. So what's going to happen is instead of 2.5 centimeters long, it's going to be 1.25 centimeters long, 1.25 centimeters high, and 1.25 centimeters wide. So this way it's going to be a total of 8 blocks, but if you add up the volume for both of these, okay, it's actually going to be the same. So when you calculate the surface area, just as a review, is you take the surface area of each side, it's length times width, and that's the surface area of the side, and because it is 6 sides, you got to times it by 6. To get the volume, length times width times height, or side cubed. On over here, this is the so this is one sample. The second sample that I want you to consider is all of these blocks together as one. So the volume for all the blocks is length times width times height of each one times 8. You're going to find that it's the same volume, it's the same material. You should get the same number as on the left. The surface area is going to change, however. It is going to be the side of the length times the width, and that's going to give you the surface area of one side. But what's going to happen, it is going to be times 6. Then, after we times it by 6, we need to times that by the 8 cubes. So you're definitely going to get a different surface area. Finally, what you have to do is you have to calculate the surface area to volume ratio. So to do that, you're going to take the surface area number, you're going to divide it by the volume. When you divide those numbers, you're going to get an actual number. That is going to be the number that represents the ratio. So when you get this, I want you to observe that the large block has a specific surface area to volume ratio, and all of the small blocks have a different surface area to volume ratio, and we will get to that at the end. So when we put it in the vinegar, we want to time how long it is going to take for the vinegar to penetrate into the agar. How are we going to know that? Well, just like before, we saw that the vinegar neutralized the sodium hydroxide and it turned it from pink to clear. We are going to look to see how far the clear coloring goes in. All right, so we put it in the blocks. After 5 minutes, we could all actually see the clear coloring already starting to appear, most pronounced in these in the block that's cut up into the small pieces right here. Don't forget these were the same size block. We just cut this up into 8 smaller pieces. Okay, and then after 10 minutes goes by, it is really pronounced in the small ones, and we start to see it in the large block that's uncut. After 15 minutes, we can see that it's even more pronounced. So it almost looks like a little box within a box. It's kind of cool looking. In real life, without looking at the picture, it's even better. And after 20 minutes, this is pretty much gone. So after 20 minutes, you can count this as completely gone, pretty much in all of the cut agar. But look how much is still left. You could actually see how much is left there. And what's going to happen is after 25 minutes, it's really starting to disappear. But you can still see in the large one, it's still in the center there. Without the picture, in real life, is even more pronounced. And then after 30 minutes, it is completely gone also from the larger one. So if you want to record the data, it's 30 minutes for the uncut block, and it was about 20 to 25 minutes for the smaller the block cut up into small pieces. All right. So why did it take faster for the diffusion to occur in the block cut up into small pieces? Well, one of the main ideas of why cells are so small is to increase the surface area to volume ratio. So that's when you take the surface area divided by volume. So both of these have the same amount of cellular content. So if you would imagine these as cells, the amount of cytoplasm that includes the chemical reactions, it includes all the activities, is inside here. So same amount of material. However, oxygen needs to come in. Carbon dioxide needs to leave. Nutrients need to come in. Stuff needs to diffuse back and forth. Ask yourself the question, when is it actually more efficient? When is it going to be diffusing at a faster rate? After you answer that question, you could then begin to really think about how efficient a small object is at exchanging material by diffusion. It actually goes at a faster rate. So there are other ways that cells can increase the surface area. One is that it can divide to get smaller, also change its shape. So what kind of shapes do you think, or can you come up with, that could increase the surface area, keeping the volume the same? Even if you take one small block individually, right, and calculate the surface area volume ratio of just one of the small pieces, it actually is still much larger than the large block. So you should do that also when you're working with these numbers. So I hope that helped, and I hope that you understand the lab, and I shall talk to you guys next time.
Thank you. Okay, so that is an experiment to prove that having a larger surface area to volume ratio is beneficial for cells. Okay, let's just quickly run through this. So, okay, so you can see, right, for cell A, this cell can obtain food and oxygen from all of its sites. For cell B, it has the same proportion, okay, blah blah blah. Basically, it can only obtain half the amount of food and oxygen per unit time as compared to the previous cell. And for cell C, it's even smaller surface area to per unit volume, and so you get even less food and oxygen per unit time as compared to the previous two. So this cell is the least active as compared to the other two cells. Okay, so there are different ways, like mentioned in the video, that the cell can actually even increase their shape to increase the surface area by changing their shape. Okay, so one is by spreading itself out. How do they do that? Okay, so firstly, they can elongate themselves to form more of a rectangular shape. And this is similar to the cells that you see in your root hair cell, right? They have that long and narrow extension. And also in the cells that are inside your small intestine, these are called the microvilli. They have also long and narrow, this time we call it protrusions. And basically by doing that, they increase their surface area whilst keeping their volume the same. So let's say cell A is a square, it has a surface area of 6 and a volume of 1, so it's 6 to 1. Whereas in cell B has a surface area of 10, the volume of 1, so 10 to 1. So cell B wins because it has a higher surface area to volume ratio. Let's look at some specific examples. Okay, so some cells are specifically adapted to absorb substances. For example, the root hair cell in plants and the epithelial cells of the small intestine, which I just mentioned briefly just now. These cells have long and narrow protrusions or folds in the membrane. This will actually increase the surface area to volume ratio of the cell membrane through which materials can be absorbed into the cell. Okay, we will learn more about these particular cells in chapter 5, but just to let you know now that in your small intestine is lined with epithelial cells. On these epithelial cells, you have hair-like or finger-like objects called villi. These are called this one whole structure is called a villi, right? That's all these cells together. It's called a villi, and inside each of the cells you have a microvilli. Okay, so it's like a villi on the villi, microvilli. And these microvilli are there specifically to increase the surface area to volume ratio of the small intestine, so that you can increase the rate of absorption of nutrients into the bloodstream. Okay, this over here, this red and blue part, this is actually your bloodstream. The capillaries are actually carrying your blood in. They're absorbing the food that's absorbed will go into the capillaries. Some of them will also go into this thing called a lacteal, and we'll talk about it in the future. And then they'll be carried away to the other parts of the body. Okay, so this is yeah, so basically that was the surface area to volume ratio. Okay. That's not. Okay, let's look at these comments. Okay, so one person says only the water molecules will move as they have enough energy. Okay, so they're looking at this T, right? Then another man says the T molecules will stay where they are and not move until they gain enough energy from the hot water. Okay, sure. And C says both the T molecules and water molecules will move about in the pots. Okay, so who do you think is correct? Let's look at some hints. Firstly, why is the color of the tea darker in the region near the tea bag when the tea bag is first placed into the water? And what will the color of the tea be after 1 hour? Okay, so we've taught in chemistry, we learned about this thing called kinetic particle theory. Basically, when the temperature increases, particles will gain more energy and move faster, so the rate of diffusion increases as the temperature of the solution increases. So answer is actually C is correct. Okay, why is that? So the water, right, is hot, so it's moving around, and as it moves around, it also increases the energy of the T molecules. So the T molecules will then start to diffuse out of the tea bag. Now, you know the tea bag is, the holes are big, so that is basically a permeable membrane. The tea bag is a permeable membrane. It doesn't restrict the movement of the T molecules. So the T molecules will then move around until they also diffuse through the entire pot of water, and there is a equal concentration of T molecules throughout the entire pot. And so this is why it requires an equilibrium of molecules. Okay, can you take some time to do these questions? 1, 2, 3. Okay, let's pause here and I will do part 2 for you soon.